welcome to the Dr. Naaz Show. Today our topic will be protein synthesis with our guests Dr. Isabel Ermack, Dr. Professor Petra Silverman, and our specialist, Miss Alexis Treese. Let's introduce our first guest today, Dr. Isabel Ermack, DNA specialist from UC Berkeley.
Nice to meet you.
Nice to meet you. So tell us, Dr. Ermack, tell us the basics that we need to know about your study.
Well, you probably don't realize it, but your hair, your eyes, you are you because of DNA. But you might be wondering, what determines if one person has blonde hair or brown? The answer can be found by looking closely at the underlying processes and functions of DNA genes and proteins. DNA organizes into units called chromosomes, and these units make up your genes.
But how does this relate to protein synthesis?
Well, your genetic code codes for specific proteins that are produced during protein synthesis and which allow for certain traits to be expressed in individuals.
Wow, so you're telling me that I am a product of DNA?
Yup, fascinating, isn't it?
Quite. Thank you for joining us today, Dr. Ermack.
Thank you for having me.
You're very welcome. Our next guest is Dr. Professor Petra Silverman, #1 amino acid and codon specialist from UCLA. She has worked with countless celebrities in the Beverly Hills area. Let's give another warm welcome for Ms. Petra Silverman. Can you tell us a bit about your work with amino acids, Ms. Silverman?
Dr. Professor, if I must. Well, amino acids are what make up polypeptides, and polypeptides are what make up proteins.
Oh, well, what types of celebrities have you worked with that agree with this amino acid trend?
Well, Miley Cyrus is one of my biggest fans at the moment, because both her and I agree that if you can't understand basic biological functions, then how are you supposed to keep up with Hollywood trends?
Fascinating. How do these amino acids relate to protein synthesis?
My work's always fascinating, but if you insist. In protein synthesis, amino acids have to be assembled in accordance to the genetic codes found on DNA, which is then transferred onto mRNA. There is an amino acid codon chart that tells which codons match which amino acids.
Wow. Can you explain to us a bit more about these codons and anticodons you work with?
Yeah. Well, the codons are a 3-base part of the mRNA, which have to match up with the anticodon, which is located on one end of the tRNA, while the other end of the tRNA holds an amino acid. The codons and anticodons have to sort of fit together like a puzzle piece, per se.
Wow. Thank you so much for being here today, Ms. Silverman. Dr. Professor Silverman.
Mm-hmm.
Nice. Our final guest today is the #1 ribonucleic acid and transcription / translation specialist from the University of Chattanooga in Tennessee, Dr. Alexis Treese.
Hi.
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 Nice to meet you.
Nice to meet you.
So tell us, Ms. Treese, what exactly are ribonucleic acids?
I just want to make sure that it's okay that I bring my notes. Mom always said I wasn't the sharpest tool in the shed, you know?
Oh, that's fine.
All right. So what was your question?
But tell us, what exactly is a ribonucleic acid?
A ribonucleic acid is a family of large biological molecules that perform multiple vital roles in the coding, decoding, regulation, and expression of genes. Like DNA, RNA is assembled of a chain of nucleotides, but is single-stranded.
Wow, interesting. So, what specific roles do these RNA molecules play?
Well, there's mRNA, which is the messenger RNA, just like your email. All the genetic information about how to produce a new polypeptide chain or a protein strain will be copied to the mRNA. Then it will move to cytoplasm and attach it to the ribosome, which is the factory to produce the protein. tRNA is a transfer RNA. tRNA is a function mainly in transferring the mRNA. rRNA is the ribosomal RNA. This is the site where the protein is synthesized. At this site, all the genetic info copied by mRNA will be translated, and a new strain of DNA will have been produced.
Wow. Can you tell us about your work in transcription and translation?
This is my favorite. Transcription is the first step of a gene expression in which the particular segment of a DNA is copied into RNA by the enzyme RNA polymerase. Both RNA and DNA are nucleic acids. In molecular biology and genetics, translation is the process in which cellular ribosomes create proteins.
Wow, thank you for all of that information, Ms. Treese. Very nice to meet you.
You too. Thank you for having me.
You're welcome. You're welcome.
I've always wanted to be, in the big city. So.
Yeah, we can see that.
Yeah.
Anyway.
All right.
Bye.
See you later.
Uncut. So in conclusion to our show today, protein synthesis consists of 20 different amino acids that make up most proteins, enzymes, DNA, and RNA. The protein is synthesized in the cytoplasm after the RNA carries instructions from the nuclear DNA. The 4 types of RNA that function in the protein synthesis are the ribosomal RNA, the transfer RNA, the messenger RNA, and the regulatory RNA. Protein synthesis begins with the process called transcription, in which a strand or the messenger RNA is synthesized according to the nitrogenous base code of DNA, and ends with translation, in which is the process where cellular ribosomes are created. I would also like to give a big thank you today to our guests, to our guests, and to viewers like you. Thank you.